everyone, welcome to my channel Biologics 5, and today we are going to discuss microbes in human welfare. As we all know, microbes are our friend as well as our foe. They cause different diseases in different organisms and they are our friend because there are so many beneficial effects of microbes in our daily life. So in this chapter we will discuss only the beneficial effects of microbes or the use of microbes in human welfare. Now comes to the microbes. What are microbes? Actually these are tiny organisms which we can't see through our naked eye. These are only visible under microscope. That's why they are called microbes or microorganisms. They are visible under microscope. These are different type of microbes: bacteria, viruses, fungi, algae, protozoan, and these archaebacteria. And they are present everywhere in the atmosphere, in biological organisms. That's why they are called omnipresent, present everywhere, in water bodies, present everywhere. Some of them are present even at the sites where nobody can survive. These archaebacteria can survive at 100 degrees centigrade, also in thermal vents, and they can survive under several layers of snow or ice at 190 degrees centigrade, minus 190 degrees centigrade. So they can survive in harsh conditions where nobody can survive. So that's why these are omnipresent, present everywhere. Now come to these petri dishes. These are the petri dishes in which different fungal colony, bacterial colony are growing. We can't see them individually through the without the microscope, but we can see these colonies of bacteria with our naked eye. These are different colonies of bacteria which we can see our naked eye, and each colony contains millions of bacteria. That's why they are visible together in the colony. We can't see them individually. And this is the fungal colony. The fungus is growing in petri dish in the laboratory for different studies. These are the fungal colony. Now comes to the use of microbes in household products. First we come to the dairy product. We consume lot of dairy products made through the activity of microbes, like curd. We use curd in different products and we use it as simple. We make curd rice, we make different ratas through curd, and these are probiotics which we consume for our good health. This is the yogurt, different product of milk made through the fermentation, made through the activity of different microbes. What these microbes cause to the milk? Actually these microbes are lactic acid bacteria which we utilize in forming these dairy products like curd, yogurt and probiotic drinks. What the lactic acid bacteria do? Actually this lactic acid bacteria convert the lactose sugar of milk into the lactic acid, and this lactic acid cause the coagulation of, because this is acid and it's coagulate the milk protein casein, which is present in the milk. Protein casein, that is the protein of milk. Casein, it coagulate milk protein casein, and the different products are formed like curd, probiotics and yogurt. In our homes we generally form or made curd with the help of inoculum. Inoculum means small amount of curd. We mixed it with the milk and keep it for several hours at warm place, and this curd is formed due to the activity of lactic acid bacteria. These lactic acid bacteria, and the important one is lactobacillus. Streptococcus is also lactic acid bacteria, and we generally in this product lactobacillus is utilized in the formation of curd. Streptococcus we use in making of yogurt. So these are lactic acid bacteria and quite important for forming different dairy products. It also increase This lactic acid bacteria also increase the nutritional quality of product. Nutritional value of product is increased because the concentration of vitamin B12 is much more in these products like curd, probiotics in comparison to milk. So nutritional value of these products increase due to the activity of these lactic acid bacteria. Next we come to the another product, that is cheese. The yummy cheese, different dishes we generally like. This cheese, what is actually this cheese? It's a concentrate of milk, fat and protein casein. What is this cheese? This is a degraded concentrate, partially degraded, and this is a partially degraded concentrate of milk, fat and protein, that is casein. And how it is formed? It is formed by the activity of different microbes. Different variety of cheese are available in the market. We know that there are different type of cheese: mozzarella cheese, Swiss cheese, Roquefort cheese, cottage cheese, and different variety of cheese depend upon their specific texture, flavor and taste, and how these are different from each other because the microbes used for making them are different. Different cheese are formed due to the activity of different microbes. First we come to the Swiss cheese. Here we have the picture of Swiss cheese. You can see here. This is characterized by these large holes and a specific texture. These large holes are formed due to the amount of CO2 released by the bacteria used for its ripening. And which bacteria we use for this ripening of Swiss cheese is Propionibacterium shermani. This is quite important bacteria which we use for ripening this Swiss cheese, and the large amount of CO2 released which form these holes in this cheese. Next we come to the Roquefort cheese. The microbe which we use for ripening this cheese is Penicillium roqueforti. It's a fungi actually, and this is made up of sheep milk, and it is characterized by, you can clearly see here in Roquefort cheese that there is a green blue mottling over there on the cheese block. This blue green mottling is due to the activity of this Penicillium roqueforti, and due to this mottling, green blue mottling, it is also known as blue cheese. So this is Roquefort cheese made by the activity of Penicillium roqueforti. Next we come to a different product, other product, that is bread. We use microbes in bread making, and the yeast we use, yeast, it's a fungi, unicellular fungi, Saccharomyces cerevisiae. We use it in bread making. We mix the flour with this cheese and knead it and kept it for some hours, and after some hours it puffed up due to the release of carbon dioxide. So this microbe Saccharomyces cerevisiae is also known as baker's yeast because it is quite important in the industry of baking. That's why it is known as baker's yeast. That is Saccharomyces cerevisiae, and the bread we form used in different type of products. In pizza we use bread, and in cake making we need bread. So bread is used in wide level, at wide level. So next come to the batter we use for making different dishes like dosa, idli or jalebi. Actually these all dishes are formed by the fermented batter. We mix the batter and kept it for overnight for the fermentation, and for fermentation microbes are required, and these microbes are these Leuconostoc mesenteroids and Streptococcus faecalis. Actually these are the microbes which is already present in the atmosphere. When we kept the batter for fermentation, these microbes cause fermentation of batter and release CO2. The puffed appearance of batter is due to the activity of these microbes. These are actually lactic acid bacteria. These are lactic acid bacteria, Leuconostoc mesenteroids and Streptococcus faecalis. So different dishes are made by this fermented batter. which are quite yummy. We all liked it. Next, come to the drink, that is the toddy drink. It's a refreshing drink which is quite popular in the southern India. Toddy drink, refreshing drink. How it is made? It also made by the activity of microbes. Here we ferment. What we ferment? Latex obtained from toddy palm. We collect the latex from toddy palm and mix with coconut water and let it ferment. After fermentation, we get the drink, toddy drink, that is a quite refreshing drink in South India. We can further make jaggery with this drink by heating this drink, make jaggery. The next important use of microbes is at industrial level. Use of microbes in industry. In industry, we use microbes at quite large scale. At industrial level, we produced different products like antibiotics and fermented beverages and antibiotics on large scale. Large scale means in large amount, and for which we require these fermenters. Actually, we required these products in large amounts, so we use large vessels in which fermentation going on with the activity of microbes, and the products we get generally are fermented beverages and antibiotics. These fermented beverages include actually alcoholic drink like rum, whiskey, vodka, etc. Antibiotics are quite important because they are life saving. Now come to our fermented beverages. We use different microbes in them, and here in fermented beverages we use brewer's yeast, because in brewing industry we use different species of Saccharomyces. This is Saccharomyces cerevisiae. This is a yeast, Saccharomyces ellipsoides, and others also. So these collectively, these species are known as brewer's yeast, which we used in this fermentation of different beverages. And the drinks which we get through the activity of fermentation, we ferment. What we ferment? Actually malted cereals and fruits. These fruits, juices and malted cereals are fermented by the activity of this yeast, that is Saccharomyces cerevisiae ellipsoides, and we get the fermented beverages. In the fermented beverages we get from the activity of these microbes are wine and beers and whiskey, rum, brandy, vodka, etc. How? These are little bit different because the activity of different microbes. Wine and beers are actually produced without distillation. This is the process distillation which we use for formation of different drinks. Through distillation we get the product. By using distillation, by doing distillation, we get these products. Actually they are purified little bit and have a higher amount of alcohol. These products are higher amount of alcohol, and they are also known as hard liquors because they have high amount of alcohol and produced through the process of distillation. These drinks, wine and beers, are obtained without the process of distillation. They have lesser amount of alcohol, wine and beer. They have high amount of alcohol and called hard liquors. Next come to the antibiotics. This is the most important products which we get from the microbes, antibiotics, because they are life saving. They are used against different diseases. Antibiotics means against life. Against life of what? Pathogen, not for ours. They are against life of pathogen. They kill or inhibit the growth of pathogen by their activity, and they are secreted in a very small amount. So the antibiotics are chemical substance produced by microorganism in very small concentration which inhibits the growth or kill the pathogenic organism and save us from different diseases without harming the horse. They don't harm us, actually they save our life and they are pro-life for us. The first antibiotics which is discovered was penicillin. And who discovered it? Discovered scientist is Alexander Fleming, quite important scientist Alexander Fleming. He discovered first antibiotic, and that is penicillin. And from which fungus it extracted? Penicillium notatum. And this is a by chance discovery. Why it is called by chance discovery? Because Alexander Fleming is working on different project and he got different thing. He is working on actually Staphylococcus. Alexander Fleming is working on Staphylococcus, a bacteria, disease causing bacteria. And one day Alexander Fleming observed that in some unwashed dishes this fungus is growing. This fungus is growing and it is inhibiting the growth of Staphylococcus. That means he concluded that this fungus secreting some substance which is against the growth of Staphylococcus. So he discovered this chemical and named it as penicillin. It was commercially extracted by the efforts of Chain and Florey, the biochemists who extracted penicillin at the commercial level. And this penicillin is extensively used in treating wounded American soldiers in World War II. That's why it's quite important, saved life of many soldiers, penicillin. And then Alexander Fleming, Chain and Florey together were awarded with Nobel Prize in 1945. So it was quite important, penicillin discovery. These are different antibiotics. List of different antibiotics like streptomycin, chloramphenicol, chloromycetin, tetracycline, penicillin, bacitracin. These are used against different diseases. For typhoid, typhus, whooping cough, pneumonia, tuberculosis. So these are quite important. These are different source from which these antibiotics are extracted. Actually these are microbes from which these antibiotics are extracted, and these are the actions of these antibiotics against different diseases. So this is all about the microbes, use of microbes at industrial level. Now we come to the third topic, and that is chemical enzymes and bioactive molecules we obtain from the microbes. Actually these are different chemicals like organic acids which we get from these microbes. These are the source of these acids. Citric acid we get from the Aspergillus niger fungi. The acetic acid we got from the Acetobacter aceti. Butyric acid from the Clostridium botulicum, and lactic acid from lactic. We have to learn actually these names for exams. These are important organic acids, and these are the different enzymes which we get from the microbes and their uses. First enzyme comes the lipases. Lipase means lipid dissolving enzymes. So these are lipid dissolving enzymes, and the source from which microbe we get it, Candida lipolytica and Geotrichum candidum. So these are the source of lipases, and the use of lipases actually the lipid dissolving enzymes. So we use them in the detergent industry. So in making detergents we use lipases, and these are helpful in removing all stains from the laundry. So quite important enzyme. Next enzyme we get from the microbes, pectinase. And this pectinase, what it will do? With the help of proteases, these pectinases used in clarifying fruit juices. The juices which we made in a home are little bit turbid. I mean they are not transparent as maki juices. So this pectinase enzyme along with proteases are used for clarifying juices. The fibers from these dissolve to the help of these enzymes. Next we come to the streptokinase, quite important enzyme. We obtain it from the Streptococcus. It's a bacteria, and it is used as a clot buster. The Streptococcus is used as a clot buster inside the blood vessels of heart patients. Sometimes in heart patients the clot is formed, and this clot can be removed through the help of this enzyme streptokinase. Quite important streptokinase. And this is the bacteria Streptococcus from which we can obtain this enzyme. Next, come to the bioactive molecule. Here, this is the cyclosporin A. This is a bioactive molecule. We obtain it from Trichoderma polysporum. The microbes from which we get these bioactive molecules are quite important. We have to learn the name of these microbes for our studies. Cyclosporin A we obtain from Trichoderma polysporum, and it's an immunosuppressive agent. Immunosuppressive agent suppress the immune system. Actually, during transplantation, what happened? The organ we transplanted was rejected by the body. So, we have to suppress the immune system to avoid the rejection of the organ transplantation. So, we use it as an immunosuppressive agent during the organ transplantation. Next is a statin. Statin, we get it from the Monascus purpurus. It used as a blood cholesterol lowering agent by competitive inhibition. So, process competitive inhibition. Through competitive inhibition, this statin work as competitor of enzyme required for the formation of blood cholesterol. So, this statin function as a competitive inhibitor of enzyme, and it inhibit the activity of enzyme used important for the formation of blood cholesterol. So, it lowers the level of cholesterol. These are all about the bioactive molecule. We have discussed all these topics: microbes in household products, microbes in industry, and these chemical enzymes and bioactive molecules. Hope you guys understood these topics well. And if you like my video, please subscribe my channel and ring the bell if you want to get notified whenever